Namaste everyone, I'm your co-host Shravani ma'am. Welcome to your favorite learning channel. The topic for today's video is microscope. So to begin with, we would see what is a microscope. Okay, so we would begin with the introduction. Okay, so first we would understand the introduction of microscopes. That is, what is a microscope? We must be knowing. So what is microscope or what are microscopes? They are optical instruments. What are they? They are the optical instruments. Optical instruments, and these instruments are used for viewing. Viewing of what? Viewing the small objects that cannot be visualized. That cannot be visualized by our naked eyes. Naked eyes. So what are the microscopes? These are optical instruments that are used for viewing small objects that cannot be visualized by our naked eyes. Okay, so what are these small objects? These small objects could be cells, microorganisms which cannot be visualized by our naked eyes. So in order to visualize them, we use these optical instruments known as microscopes. Okay, so now let's see or talk about some history of microscopes. History of microscopes. Okay, so here, so with the advent of microscope, this advent of microscopes actually helped in the discovery of cell. So the first microscope was constructed by whom? The first microscope was constructed. It was constructed by the scientist named Anton van Leeuwenhoek. Okay, Leeuwenhoek. So he was the person who constructed the first microscope, and it is called the simple microscope. Simple microscope. Okay, so this simple microscope has an eyepiece which is located very close to the lens on one side, and the object can be placed on the screw point on the opposite side of the lens. Okay, this is about simple microscope that was first constructed by Anton van Leeuwenhoek. After that, what happened? So with this simple microscope, Leeuwenhoek could magnify and view objects up to 200 times. Later on, Robert Hooke developed, what? He developed a compound microscope. So we would study about compound microscope in detail in our next section. Okay, so Robert Hooke later developed this compound microscope. Okay, now let's see what are the major types of microscopes. So the major types of microscopes. So what are the major types of microscopes? There are actually three major types of microscopes. Okay, what are they? One is the compound microscope, compound microscope. Then there is light microscope, and the other one is the electron microscope. Alright, so we would study about each of them in detail now. First is compound microscope. Just now we saw that after the discovery or the construction of simple microscope by Anton van Leeuwenhoek, Robert Hooke had constructed or developed a compound microscope. Okay, so this compound microscope, how was it developed? So Robert Hooke developed a compound microscope using two lenses. Okay, it was developed by Robert Hooke, we already know, by using how many lenses? Two lenses. Okay, so as to observe maximum magnification. To observe maximum magnification. Okay, so in this microscope, what happens is, in this microscope, the object is placed on the stage. Here the object is placed on the stage that is present below what? That is present below, and what happens to the light? So the light that comes from an oil flame was thrown on it by what? By a convex mirror. Okay, so Robert Hooke developed the compound microscope by using two lenses so that he could observe maximum magnification, and in this microscope the object is actually placed on the stage that is present below, and light that is coming from oil flame was thrown on it by a convex mirror. Okay, so this is a compound microscope. Then the next one is light microscope, or it can also be called as optical microscope. Okay, so it is greatly improved design of Hooke's microscope. Okay, this is an improved version of Hooke's compound microscope. So it has two lenses. It consists of, or it has two lenses. What are these two lenses? Eyepiece lens. One is called eyepiece lens, and the other one is called the objective lens. Now what happens is, these two lenses are combined to produce a greater magnification. Okay, so the light microscope shows magnification up to, what is the magnification shown by light microscopes? It can show a magnification up to 1500 times, which is good enough to see. This is good enough to see or visualize what? The cells also the larger organelles, etc. Okay, this type of magnification is actually good enough to see the cell's larger organelles, etc. Now let's talk about the next type of microscope, that is an electron microscope. Electron microscope. So electron microscope has the powers of magnification and resolution greater than an optical microscope. It has the powers of magnification and the resolution of electron microscope is greater than what? Than an optical microscope. Okay, so in this microscope, a beam of electrons, what happens here? A beam of electrons is passed. It is passed through what? It is passed through the section of the material, okay, to produce, in order to produce what? In order to produce the image. Okay, so this is about electron microscopes. Alright, so now let's quickly revise everything that we have covered today. So today we spoke about the microscopes. So now we know that microscopes are optical instruments which are meant for viewing small objects that cannot be visualized by our naked eyes, like the cells, microorganisms, etc. Okay, so then we talked about the history of microscopes, that who discovered microscopes at first. Okay, so the first microscope was constructed by Anton van Leeuwenhoek, which is the simple microscope. So what happens in simple microscope? The simple microscope has an eyepiece that is located very close to the lens on one side, and the object can be placed on the screw point on the opposite side of the lens. Okay, so at one side there is lens, and on the opposite side, on the screw point on the opposite side of the lens, the object to be visualized can be placed. This is in case of simple microscope, which was first constructed by Anton van Leeuwenhoek. After this, Robert Hooke constructed a little more advanced form of microscope called the compound microscope. Then we talked about the major types of microscopes. So there are three major types of microscopes. One is the compound microscope, then there is light microscope, then there is electron microscope. Okay, so this compound microscope, as we already discussed, it was first developed by Robert Hooke by using two lenses so as to observe the maximum magnification. So here what he did was, the object was placed on a stage below, and the light coming from an oil flame was thrown onto this object by a convex mirror so as to obtain maximum magnification. Okay, then we saw what was light microscope or optical microscope. So this light or optical microscope is an improved design of Hooke's microscope. So it has two lenses, one is the eyepiece lens and the other one is the objective lens. So what happens here is, these two lenses are combined to produce a greater magnification, and this light or optical microscope shows magnification up to 1500 times, which is good enough to visualize the cells, larger organelles, etc. Okay, then we also learned about the electron microscope. So The electron microscope has powers of magnification and resolution that is greater than an optical microscope. So what happens here? Here a beam of electrons is actually passed through the section of material to produce the image. Okay, so here the beam of electrons is used in order to produce the image which is passed through the section of material. Okay. So I hope you all would have got some great insights by watching today's video. Please do not forget to like and share the video. Till then, thank you for watching.